In Chapter 13, now we're going to talk about how to control the growth of microorganisms. It's kind of amazing sometimes when you look at different items and then you do a swab on it and you figure out what is growing on there. Sometimes I guess it's to an advantage that we can't see individual microorganisms because they're so small, so we don't see how highly contaminated things are. Sometimes people will probably say ignorance is bliss. This is a study looking at several different cars and trying to determine how many bacteria are in certain things, such as on the door latch. As you can see here, the inside of the steering wheel had quite a number of bacteria on them. The center console, look at this number here. I guess because we're constantly putting food and drink, we're constantly touching along the center console. I guess very few people are touching the window control unit, so it just gives you an idea of microorganisms are all around us. So how do we control this growth? Before we understand how to control the growth, a couple of things to be aware of. Oftentimes microorganisms are transmitted from one location to another or from one person to another by inanimate objects, and we refer to those as fomites. The other thing is with labs, not all labs are the same. And so we have different levels that we classify them as. And that determines how you dress in them. It determines which organisms you can work with in them. It all has to do with safety. So we classify labs as biosafety level 1, 2, 3, or 4. A biosafety level 1 or a BSL1 lab. In this type of a lab, you can work with non-pathogenic organisms. You have your standard PPE, personal protection, things such as gloves, maybe a lab coat. In a BSL level 2, you're working with what are classified as low-level pathogens. For most people, it shouldn't make you sick, but you still are going to wear your standard protective personal protection device. You're going to also work in what's known as safety cabinets or in hoods. So you're not necessarily going to be working with the bacteria with their petri dishes and your test tubes out on an open lab bench. You would in a BSL1, but not in 2. You're going to be working in a special, when we say a safety cabinet, it's often a laminar flow hood where there's a special airflow to always be blowing the air away from you to try to prevent contamination of you or contamination of your samples. In a BSL3, now you're working with very serious pathogens. These could be potentially fatal. And so when you're working with them, you need to be wearing a respirator. Of course, you're going to have your lab coat, your gloves on, safety goggles, all of that. But in addition, you'll be wearing a respirator. Usually there's limited access. Only authorized personnel only are allowed into those labs. BSL4, this is where you're working with the worst of the worst. You're working with the most dangerous pathogens here. These are the ones that definitely can kill you. And there may be no cure for you if you do get sick. It'd be, I'm sorry, there's nothing we can do. There are only a couple of labs in the United States that are rated as a BSL4. So obviously they're not very common. You have to be specially trained to work in them. You are wearing a full body protective gear. Obviously separate respirator airflow. You go through a process just to enter the lab, and the entrance to a lab is very tightly controlled and restricted. So when you walk into it, you go into a room where you change, you put on your suit that you wear, your protective gear, and from there then you go finally into the lab. You work in the lab. When you go to leave, you have to go through a decontamination process. You don't walk out the door into the hallway. There's a series of rooms you go through, decontamination to then a special way even of stepping out of the protective gear. All this is to help keep you from getting contaminated. like I say, you go through this decontamination process prior to exiting the lab. Most of the time in a BSL-4, you do not work your 8-hour shift in the lab. It's very stressful. The protective gear gets heavy, and so usually you work kind of in shifts, only a few hours at a time. And here is a picture of two individuals getting ready to go work into a BSL level 4. So you can see they're in full body protective gear. These coil tubes that you can see, he's attaching it to himself. This goes up to the ceiling, and that is attached to your air supply. And so you are walking around, you are never exposed to the atmosphere in the lab itself, because you may be working with airborne pathogens. And like I say, you're working with the worst of the worst. And so you attach this to your suit. Sometimes these are on little trackways on the ceiling, and so if you have to go a great distance, you may have to disconnect in one place and then go reconnect to a new tubing to get further distance. The CDC in Atlanta is Center for Disease Control. That is one of the labs that has a BSL-4 rating. So, like I said, the CDC is going to classify all microorganisms into what the risk factor is and how you can work, and which type of lab do you work with them in. And so, like I say, non-pathogenic strains, or a BSL-1. Things that would be a moderate risk to the workers. Usually, as long as you're a healthy individual, you're okay. Things like Staph aureus. BSL-3, more serious pathogens, such as Mycobacterium tuberculosis. And then the BSL-4, things where there's no known cure. Ebola. I remember years ago when I was living in New Mexico, when they first were in the process of identifying hantavirus. They worked with that, and it was sent to the CDC lab in Atlanta, and that's where they worked, because all they knew at that point, before they identified it, was they had something that was, you had individuals in their 20s who were perfectly healthy, and a week later they were dead. So you knew it was something very serious. So you don't want to take a chance until you know what you're working with. You don't work with it in an open lab. So these are the safety levels. So when we talk about, okay, how do we control the growth then of the microorganisms? We're going to be very picky on our definitions here. And you might think, oh, you're kind of strict on this. Yes, we are, because to us as microbiologists, it makes a difference. Sanitation and sterilization are not the same thing. To a microbiologist, sterilization means that you have killed everything. You've killed all the microorganisms, all the endospores. There is nothing living in that on that utensil, in that sample, in that test tube, or whatever. It is sterile. There is nothing growing on it. How do you carry out sterilization techniques? You can use physical methods or chemical methods. Physical methods, usually it's going to involve high heat, high pressure, and filtration. Chemical methods, to sterilize things, you have to expose it to a chemical for the appropriate amount of time. And that amount of time, it's going to depend on which chemical you're talking about. So like I say, our vocabulary is a little picky here. To control the growth, one thing you use is aseptic technique. That helps to prevent contamination on sterile items or within a sterile area. In any microbiology lab, you learn your sterile technique. Those of you going on to various careers in the healthcare field, they will also teach you, how do you put on your gloves properly? Certainly if you're going, let's say, into surg tech, how do you keep your instruments sterile for the doctor to use to perform the surgery? You don't want to introduce any infections. So you will learn the techniques as to how to handle things to keep things sterile. In a microbiology lab, when we talk about aseptic technique, we're usually talking about working around your flame, keeping within 6 to 8 inches of that open flame. You move further away from that, you're now outside your sterile area. You need to redo your technique of sterilizing something. When we say we're disinfecting something versus antiseptics, so here goes some of our definitions. A disinfectant is going to kill most, but not all microorganisms, but it's on an inanimate item. You can disinfect the tabletop. An antiseptic will kill most, but not all microorganisms on living tissue. So, do you disinfect your skin? No, you would use an antiseptic on your skin. Do you use an antiseptic on a table? No, you would disinfect a tabletop. So, antiseptics with living tissue, disinfecting items with inanimate objects. Both of those, they're going to reduce what we call the load. They're going to reduce the number of microorganisms, but we also know it's not going to kill everything. It's not going to sterilize it. Degerming just means you're reducing the number of the microorganisms. When you wash your hands, that's what you're doing. You're reducing. You're not removing everything on your hands, which, by the way, you cannot sterilize your hands. Any process that will sterilize, which, as I just said, is going to kill everything, that is going to cause so much harm to your hands. You can't do that. But you can reduce the number. Sanitation is removing the load on fomites, reducing the number of microorganisms that are on. Remember, fomites are an inanimate object that can transfer microorganisms from one person to another, from one location to another. So when you sanitize something, yes, you might be in the restaurant industry. You're washing and then sanitizing, let's say, the utensils, the forks and everything else. Are you sterilizing them? No, but you're reducing the load. So what are some of the qualities of your ideal control agent? Well, you want it to be selective against pathogens. You want it to kill the pathogenic microorganisms, but not the good ones. You don't want it to cause any tissue damage on, say, your hand. If you're going to be using it on your hands, you don't want to cause any damage to your cells. You want it to act fast. You don't want to have to add something and then you have to wait an hour before it becomes effective. That's not going to help. You need it to be stable. You need it to be inexpensive. If things are expensive, people are not going to buy them. People have enough problems trying to figure out their monthly budget. If it's too expensive, they won't buy it. It needs to be easy to prepare. It also needs to be easy to use. If it's something that takes you 10 steps for it to be effective, people get bored after 2 steps and they're not going to finish it. They want it to be, let me have it cheap, easy to use, and I'm done with it. And so this just goes, there's a table just showing some of these definitions and examples of things. Another thing to keep in mind, once again, this is with our vocabulary, if it ends with cide, C-I-D-E, it means it kills the organism. So a bactericide kills bacteria. Viricide kills viruses. Fungicide kills fungi. If it says static, it just means it's inhibiting the growth. It's not killing it, but it's just inhibiting the growth. So you have bacteriostatic, virustatic, fungistatic. If it is, let's say, bacteriostatic, it's inhibiting the growth. If you change the environmental conditions, then that bacteria, you didn't kill it, so it means it could potentially start growing again. So just keep that in mind. The microbial death curve refers to what is the amount of time that's needed to kill 90% of the population. What's the length of exposure time? the susceptibility of that organism to whatever agent it is that you're testing. What's the concentration of the agent? What is the organism that you were talking about? Are you talking about bacteria? Are you talking about viruses, etc.? And so they will actually test this to see at what point is 90% of the organisms killed. Realistically, killing all of them is just sometimes not feasible, so that's why they look at 90%. So we look at both physical and chemical methods of control. In terms of physical methods, you're looking at things like heat, using an autoclave, pasteurization, freezing and refrigeration, pressure, desiccation, radiation, sonication, and filtration. So we're going to look at all of these. Heat. You have both dry heat and moist heat. Dry heat is much more effective at sterilization, but dry heat doesn't penetrate as well. Moist heat will penetrate better. So it depends on what you're working with. An example of dry heat is incineration. You do this a lot in the lab whenever you flame a loop. That's actually a form of incineration. That's a form of using dry heat. Moist heat is like boiling or autoclaving. So in this picture, in the first one here on the left, it shows you have a flame, and here is your loop, and you are flaming the end of this loop till it's nice and glowing red. That is incinerating or burning off anything that was on the end. That end of that loop will be sterile. It's also going to be hot. This is known as a bacteria incinerator or a micro incinerator, and basically here what you do is instead of a flame, you stick your loop inside this hole here. It goes all the way back here, and that's going to heat it up and incinerate it as well. An autoclave. This is used definitely with all your microbiology labs. You're using moist heat, and you're using it under pressure. So the minimum requirements for an autoclave is it must go to 121 degrees Celsius at a pressure per square inch of 15 psi, and for a minimum of 20 minutes. You have to fulfill those three things. Now you can go at a higher temperature, you can go at a higher pressure, and you can go for a longer time period. Actually, when we have a large quantity of items that we want to sterilize, we usually do run our autoclave longer than 20 minutes. But this is the minimum, 121 degrees C, 15 psi for 20 minutes. That's the absolute minimum. If you have a lot of organic material, typically you need to go a little bit longer time period as well. How do you know if your items are sterile? Is that you have some type of a quality control measure that you must use. And the easiest thing to do is using what we call autoclave tape. Now, autoclaves come in all different sizes. This is a fairly large one in a medical setting. Here at Angelina College, we currently have 2 autoclaves that we use. I have worked in a lab where my boss, I was in a water treatment lab many years ago, and my boss was so proud to buy an autoclave while I worked for him. And it's one of those situations where you're trying to act very pleased for him. Thankfully, he's starting to catch the idea that I want to sterilize things. But the autoclave he bought, you literally, to run it, you had to pour in a cup of deionized water. That was fine. But it was so small, my pipettes, I could only fit like 1 pipette at a time, and it had to be at an angle because it was so tiny. It was pretty much useless. But he was very proud of it. So most of the time what you do for quality control is you use what we just call it autoclave tape. Over here on the left, that's what it looks like initially. It looks kind of just like masking tape. If the run was successful in that it met all those minimum requirements for sterilizing things, the autoclave, it will turn with these black stripes on it. So it's a very quick, easy way. We don't necessarily put them on every single tube. We put it, if we're running test tubes, we put it on the rack. If we're running a bunch of bottles with solutions, sometimes the tub, we always put our items in a tub, just in case anything leaks or boils over. We may put the piece of tape on the tub. It just depends on what we're working with. And so when you open the autoclave, when you're done with the run, you just look at that tape. If there's a black line, yay, you were successful. If there's not a black line, okay, something happened. Maybe the seal wasn't tight on your autoclave. Maybe you didn't run it quite long enough, or the temperature didn't get it. You need to look at how your autoclave is working at that point. Pasteurization. Pasteurization is a process that was developed by Louis Pasteur. That's how it gets its name. It will kill the pathogens, but it does not sterilize. So there will still be microorganisms in the, usually you're talking about milk or dairy products. So there will still be microorganisms in that item, but they're not pathogens. The reason why you don't sterilize things like milk is that the sterilization process is going to alter the taste. It's also going to break down the protein, so it's going to change the quality. People want milk to taste like milk. And so this is, like I say, Louis Pasteur is the one that worked out the pasteurization process. There's actually a couple of different types. And so what we've learned, yes, it kills the pathogens, but it doesn't sterilize it. The traditional type of pasteurization, which is sometimes referred to as vat pasteurization, you bring the item up to 63 degrees C for 30 minutes and hold it for 30 minutes. Some of the newer methods, there's high temperature, short time, which you bring it up to 72 degrees Celsius for 15 seconds, or the ultra high temperature, you bring it up to 138 degrees C, you leave it there for at least 2 seconds. Obviously, higher temperature, shorter time period. But like I say, pasteurization will not alter the flavor. And this is just showing in large vats the types of pasteurization that's used. Here in the US, they tend to go with the high temperature, short time, the 72 for 15 seconds. And then the milk is still refrigerated. In other countries, sometimes what they will do is the ultra high temperature, because then that is packaged in airtight containers and it does not need refrigeration. So some differences there. Cold temperature tends to inhibit or slow the growth of microorganisms. So refrigeration is not going to kill the bacteria. It's just going to inhibit the growth. So something you have to keep in mind is that if you take it out of the refrigerator and say put it at a higher temperature, be that in an incubator, room temperature, whatever, is it's going to start growing again. So this is a case of where, like, bacterial static, where it inhibits the growth, but it doesn't kill it. Be aware of that, especially with food items. Please be aware of that. Ultra cold temperatures will usually kill some of the organisms. Ultra cold temperatures can be used for long-term storage, though sometimes you have to properly prepare the specimen. If there's a lot of water, when you go to freeze it, there's certain things you have to do, because when you freeze water, the water crystals can actually damage the cells. It can puncture holes in the cell membrane and ultimately end up killing the cell. So there's certain prep you need to do if you're going to do ultra cold storage. And this is showing ultra low freezer. Usually this particular one is set at -70. Some of them are set at -80, and this is degrees Celsius. And so here they're also working with ultra cold. It's a way of long-term storage, especially. Just don't, most of the freezers have alarms on them, so that if the temperature starts to go up, an alarm goes off to alert you to check it. This happened within the past year. I remember reading on the East Coast, there was at a university, and they had a freezer at -80 where they kept and stored a lot of their cultures that they had been working on for several years. That's where they kept all the kind of their backup supplies. And they had a new cleaning crew come in, and the alarm went off showing that the temperature had risen above, there's a range, and it went off. And an individual, instead of notifying someone, they unplugged the freezer. We'll just say it took them a while for anyone to realize that the freezer had been unplugged, and by that point everything had thawed out. And so we'll just say the last I heard that the university had filed a lawsuit against the cleaning company because they lost 2 years worth of research from that, which is a huge oops moment. So don't unplug them. You can use pressure to kill most organisms. What's going to happen is the cell's going to lyse, the proteins denature, and that's going to stop or inhibit any type of metabolic processes going on. So pressure over here, this is showing an example with using pressure as a means of killing things. With home canning, when you use a pressure cooker, that's what you're doing. Over here on the left, this is showing Clostridium botulinum, the causative agent of botulism, commonly found in the soil. And so this can be a problem with people who do home canning. It used to be one of the #1 ways people would get infected with botulism. So when you set up your pressure cooker, if it tells you to bring it to a certain pressure, you need to bring it to that pressure. If it says bring it to a pressure of 15, don't say, oh, 14 is close enough. No, it needs to go to 15. If it tells you to heat it for 20 minutes, you need to go for 20 minutes. Don't go for 17 and say, oh, that's close enough. No. There's a reason why somebody has worked out the temperatures, the pressure, the length of time to kill everything. These little circles in this diagram with the Clostridium, those are spores. Clostridium botulinum produces spores. Those spores are much more resistant than the vegetative cells. So you want to make sure if you're home canning, I don't know, tomatoes or strawberries or apricots or whatever it is, you don't want to can them and then later eat them and then everyone gets sick. So make sure you follow the rules. But that's what a pressure cooker is doing. It's using that pressure to help kill off any of those organisms, and especially the endospores you're worried about. Endospores are one of the hardest things to destroy. Desiccation, this is drying of the specimen. There's different ways you can do this. Lyophilization is a special form of this where you freeze dry it. This is very, very effective if you're going to do long-term storage. Basically, you freeze dry it, you store the specimen as a powder. And then when you need to use it again, you just take that powder and you rehydrate it. This sometimes is how when we order specimens from a supply company, sometimes they come in a test tube, like on a slant, but sometimes they come as a powder, and then that's what you do, is you rehydrate it. It's very effective for very long storage. You can also use high salt or high sugar because that will reduce the water content of the cells. It actually pulls water out of the cells, and cells will tend to die because of the change of the water content and the osmotic pressure. And so for food, traditionally, this has been done for hundreds of years. You have meat or you have fruit or something, you want to make jam, you add more sugar to it. That's going to reduce the contamination because that high salt or that high sugar is going to kill the organisms that are present. It's pulling the water out of the, say, bacteria. And so you have different food items here, such as making jerky or saltfish or jams, where this has been used. You can also use radiation. All these are still physical means. You can use radiation to help control the microbial growth on items. You can use either ionizing radiation, which is things like X-rays or gamma rays. It's going to interfere with the DNA structure, so it will kill the organism. The advantage of ionizing radiation, such as X-rays and gamma rays, is that it will penetrate through plastic and paper. Non-ionizing radiation also alters the DNA. However, it does not penetrate through plastic or glass. An example of this is UV light. UV light is used oftentimes in labs. I've worked in labs where when the last person leaves for the day, you turn off the lights, you switch on a UV light or a germicidal light. That's what it is a UV light. And overnight in the room, there's a UV light on there. And so it's a way of UV light will be effective in cleaning the air, the surface of the bench tops. It doesn't penetrate through the tabletops. It won't penetrate through plastic. It won't penetrate through glass. Currently in our labs are goggle cases. After every lab, when people put their eye goggles back in the case, then we lock the case up and turn on. There's a UV light in there. It's basically a germicidal case. And the UV light will kill anything that's been on the goggles. So that's the way we sterilize the goggles between classes. So this is showing how it interferes with the DNA. This shows a hood where it's got a UV light. You can see it's reflected on the bottom here because there's a UV light up on top. And then food items, now they're doing a lot of radiation, like gamma radiation. They are required in the U.S., they are required by the USDA to put the sticker on it, as you see on the right, on any item that has been treated as gamma irradiation as a means of trying to inhibit the growth of microorganisms on it. Sonication. This is exposure to ultrasonic waves. It's going to end up causing the cells to lyse or burst. Filtration, HEPA filtration. The HEPA filter is a high efficiency particulate air filter. It removes all the microorganisms from the air. You look at the pore size of it. So we put HEPA filtration filters in what's known as our laminar flow hoods, which are the safety cabinets like what you would see in a BSL-3 lab. That way any of the air that's being pulled in there is being pulled away from the lab worker to help prevent infection on the lab worker or contamination of the lab. But that air, before you pump it back outside, because we don't want everybody else to get sick, it goes through a HEPA filter. There are other types of membrane filters. We use this sometimes. I've used them. We use them currently in our lab for certain liquid solutions that contain what you call heat labile solution. Labile means it's sensitive, so it's a heat sensitive. There's a solution we work with that contains urease, you're testing for urease enzyme production, so you're looking for urea. Sometimes things with proteins are very susceptible to and sensitive to heat. And so if you're testing for certain proteins and you want a protein in the solution, heat is often going to destroy that or denature that protein. So if it's something that heat's going to destroy, you can't autoclave it. So how do you sterilize that solution? You're going to use the filtration method. Or sometimes maybe you have a solution and you want to remove the microorganisms from it. Now first, this shows the HEPA filter. It's this whole series of filters, very effective in cleaning out the air. For other membrane filters, in this apparatus on the left, you have this bottom container, which would have been previously sterilized. You connected the white is a membrane. You connect it to this upper chamber. This upper chamber is what you would pour your liquid solution into, and you've got this pump here that's going to draw a suction and pull the liquid from the top down through the filter into the bottom. But the filter is going to, you can select the pore size of the filter. It's going to leave all the dirty gunk or all the microorganisms on the top, and what's down on the bottom is sterile. Same thing, but on a smaller scale over here on the right, where you have a syringe with your liquid in it, and this has a filter in it, Once again, you buy them whatever size you want. This is what we do a lot in the lab for certain solutions, where we have sterile test tubes, which you will put the sterile test tube on the end here. You've got your solution in the syringe, just as you see here. And when you push the plunger down on the syringe, it is going to push this liquid through here. And what comes through now on this side, going into your test tube, is sterile. Like I say, you do this for heat sensitive things. So this table is just showing some of the physical methods, just a summary of it, from the heat to cold, pressure, desiccation, radiation, sonication, and filtration methods. As mentioned earlier, though, you also have chemical methods of controlling the growth of microorganisms. And here is a list of all these different types that we're going to look at. Phenolics. Phenol has a ring structure to it. They're usually very stable. They're usually very persistent. Now, phenolics are going to be a little bit less toxic than phenol itself. They will inhibit microbial growth. They will denature proteins. They will disrupt the membrane. Things like this are creosoles, things like Lysol. That's an example of one. This is showing over here under A, this is phenol, and then these are some other phenolic compounds. Trichlorosan is something that used to be in antibacterial soaps. It would inhibit the growth of microorganisms. Now, your book that we're using was published in 2016. So in your book it says that it is used. Here's an update. After the publication of your book, in the US, they did ban this in 2016. It was banned because there was growing evidence that there were some health risks that were associated with it. There were environmental risks associated with it, and they also found there was no significant health benefits to putting this in your soaps. And so whenever you add chlorine on things, that usually is a bit of a problem. A lot of your more toxic compounds do have chlorine associated with them. But this, just so you know, trichlorosan, because your book was published right before this was banned, just know that it has been banned in the United States. Heavy metals tend to inhibit the activity of enzymes, things like mercury, copper, nickel, zinc, and silver. So this is just showing some examples. Now, excuse me, some of your antiseptic mouthwashes, such as Listerine, will use zinc chloride, that the zinc is effective in killing microorganisms. Halogens, they tend to destabilize large molecules and oxidize them. Halogens are things like iodine, chlorine, fluorine. Betadine is an example. It's used as an antiseptic, commonly before surgery. Some chloramines are also good at controlling growth, like ammonia, chloramine. Alcohols tend to denature proteins. They will inhibit cell metabolism. They may disrupt the membrane. They may ultimately cause a cell to burst or lyse. Two very commonly used alcohols to control microbial growth: ethanol and isopropanol. So ethyl alcohol, yes, this is the same type of alcohol that's found in alcoholic drinks. It can also be used as a disinfectant, which is why it was fairly effective way back in Western days. And you had no other choice. Pour whiskey on the incision site before you do surgery. Isopropyl alcohol, that's rubbing alcohol. Okay. So that's used a lot. One thing I'm going to say, it's showing here with the ethyl alcohol, obviously the bottle of Perel hand sanitizer. Just be aware that a lot of your hand sanitizers do contain ethyl alcohol now, since they don't have the trichlorosan in them. anything that contains alcohol in it, that's going to dry your hands out. So just be aware, yes, it's going to kill the microorganisms, but a side effect of that, especially if you're using it a lot, this is something that some people found during the pandemic when they were using hand sanitizer a lot more than previously, was that, my hands are getting really dry, and they needed to add more lotion to it. So just be aware, if it has alcohol, it's also going to have a drying effect. Surfactants, we're talking about soaps, detergents, things like that, that will disrupt the cell membranes. So this shows what soaps have, a portion of it that's hydrophobic and then a portion that's hydrophilic. And so it's going to end up disrupting, when you use it, disrupting that membrane. I hate to tell you, but your antibacterial soaps really are not any different than just regular soap. It's the fact that it has the soap or the detergent in it. If you don't already know, those of you going into the medical or healthcare fields, you're going to learn how to wash your hands, which I'm like, I know how to wash my hands. There's a special way of washing your hands, which they will teach you when you are in the nursing program or the surge tech program or whichever one you're going into. There's a proper way of doing it. If you're going into surgery, you start with washing your hands first, and then you slowly move up to your wrist, and then your forearms, and then finally elbows. So you start at the extremity and move up. But when you're washing your hands, you need to make sure, for anyone, whether you're going into surgery or even if you're just, washing your hands, is you need to wash your hands for a minimum of 20 seconds. Okay, just passing it quickly through the water doesn't count. I'm sorry, but that does not count. So you need to get your hands wet. You need to wash. Make sure you're getting the front and the back of your hands. Make sure you're getting between your fingers. Make sure you're getting under your fingernails. And then rinse your hands off. Okay? One thing I've always said, one benefit of COVID is at least hopefully people learn to start washing their hands. We hope they watch them beforehand, but we all know not everybody does. Chlorhexidine, naloxone, or other compounds that are also going to, a lot of these you find, they help disrupt the cell membrane, which is going to lead, if you disrupt the membrane, it's going to lead to cell death. And so then the microorganism is going to end up dying. These are often used as part of your surgical scrubs as well. And then other agents, alkylating agents, they will inactivate enzymes and nucleic acids. Once again, if you inactivate an enzyme, eventually the cell's going to die. This things like formaldehyde, ethylene oxide. You don't have to know the chemical structures of these. It's just showing you how complex some of these are. Peroxygens going to cause oxidation. Things like hydrogen peroxide is probably the best known one. Ozone gas as well. Hydrogen peroxide, it's used oftentimes, number one, just on surface wounds, but also to clean some things. If you have the enzyme catalase, it's going to break hydrogen peroxide, as you see in this reaction up here. H2O2 is the hydrogen peroxide with the enzyme catalase. It'll break it down into water and oxygen. And what the oxygen will end up doing, let's say you have a wound, you were outside playing in the soil, or you stepped on a nail outside that was in the dirt. Some of the organisms like Clostridium, that's found, that is a causative agent of tetanus, that's found commonly in the soil. If you rinse that wound out with peroxide, our cells have catalase, which is why you get the bubbling when you pour peroxide on something. All that bubbling, that's the oxygen that's being produced. And in a wound, Clostridium is anaerobic, and so that oxygen is going to kill. So by pouring the peroxide on, producing the water, water's not going to do anything, but the oxygen is going to actually kill those anaerobic organisms. Supercritical gases such as carbon dioxide, what it's going to do, carbon dioxide is going to form carbonic acid, and that is an acid, so it's going to lower the pH. It's very effective at low temperatures. It doesn't change the object's integrity at all. It's non-reactive, it's non-toxic, it's non-flammable, so those are all benefits to using it. Some of your chemical food preservatives, what they will do is they will decrease the pH, and that will inhibit enzymes of microorganisms. So some of your chemical food preservatives are things like sorbic acid, benzoic acid, propionic acid, nitrates. All these, you may see them on food items at times. They are added to preserve the food. Some of your natural food preservatives, like niacin and nanomycin, are used in both cheese and meats. They inhibit cell wall synthesis. So how do you test? You can have all these various methods, whether they're physical or all the various chemical methods of controlling. How do you know whether it's effective or not? Well, by now you know we're going to rate them. We're going to organize them as to how effective they are. We organize them. We use very highly technical terms for this. We use high-level germicide, intermediate germicide, or low-level germicide. If it is a high-level germicide, if something is classified as a high-level germicide, that means it is killing everything. It's killing the vegetative cells. It's killing the fungi. It's killing all of the viruses, and it's killing those endospores. Remember I mentioned endospores are the hardest thing to kill. That's like the gold standard. If it can kill the endospores, man, that is good. So it sterilizes. It means it's killing everything. Intermediate germicides. They can kill a lot. However, they will not kill all of the viruses, and they will not kill the endospores. And then a low-level germicide, well, it can kill the vegetative cells, and it will kill some of your viruses, but it's not going to probably kill your fungi, and it's probably not going to kill the endospores. What you have to look at when deciding what you're going to use as your control agent, do you need something sterilized, or do you just need to reduce the load? What is it that you are hoping to gain? What do you need done? A lot of your high-level germicides, in order for it to kill everything, also means it's probably going to be more toxic than a low-level one. So what do you want to be exposed to? How long do you want to be exposed to something? What is your ultimate goal? You have to weigh all of this. Weigh your pros and cons. When you're looking at effectiveness of disinfectants, what's the length of exposure? What's the concentration of it, the temperature, and the pH? If you have to have something and it seems to be, hey, this looks pretty good, but the length of exposure time is 12 hours, well, that's not going to be very good. One way that you can test items is what's known as the disc diffusion method. We actually do this in lab. Students usually like doing it. You test different chemicals against a particular microorganism. And the way you do this is you take, the way we do it in the lab is we work with bacteria. You take that bacteria, you grow it on a petri dish, and you inoculate the petri dish by making what we call a bacterial lawn. So that is when you take a petri dish, and you guys are learning I'm not that great with controlling this, but you are going to cover the entire surface of this petri dish with your bacteria. That's what a lawn is. You want complete growth. You don't want to see individual colonies. You want just solid growth. So you're going to spread it. You're going to come back in all different directions. And at the very end, you're going to come around the edges. Oops, try to keep it in the plate. You're just going to want this whole thing covered. And then, before you incubate it, now what you're going to do is you are going to take these little paper discs. And literally what we use for the disc, when we hole punch papers, we keep the little shards, those little tabs, the little paper holes, and we keep them. And what you do is you're going to dip it in your chemical, and then you're going to put those little paper tabs. You can't really see this well, it's white. There we go. You're going to spread them. You can usually fit, about 4 or 5 on a plate. You're going to randomly place them around. So you might, when we do it, you might take one and dip it in Listerine, dip another one in, we usually use like Scope, Listerine, Witch Hazel, Lysol, Spick and Span. We take different chemicals, and you just dip it in it, let the excess drip off, and then you put this little paper on the surface. And then you incubate it. After you incubate it, you come back and you measure what is known as the zone of inhibition. That is around those paper discs. If there is no growth, that is your zone of inhibition. Why is it not growing? Because the chemical has inhibited the growth. So the larger the zone of inhibition, the more effective that chemical has been. And in this plate, obviously so much better than my drawings, is that, once again, you'll see why I never majored in art. And my youngest, who is an art major, obviously did not inherit those traits from me. So you can see that the solid growth on this plate, when there's growth right up to the paper disc, now I need to switch out of the black, right here where you have growth right up to that paper disc, whatever chemical was on that paper disc, it didn't inhibit the growth at all. So don't use that. Now back here you can see there's a clearing zone around this paper disc over here where they're measuring it. That clearing area, that is the zone of inhibition. On this plate, it's very easy to see. Here is a zone of inhibition. You're going to measure that distance. Here's a zone of inhibition. Here's a zone of inhibition. And even over here. The larger that zone, the more effective it is. So on this plate here on the far right, you're measuring it. It's hard to gauge just from here. But certainly either this one or this one I would use. I would not use this over here. That zone of inhibition is so much smaller, it's not as effective. And over here on the schematic drawing, you can see the same type of thing where you measure that zone of inhibition where there's no growth. The larger the area of no growth here, the more effective this particular chemical is. You can also do what's known as a use dilution test. This will test the effectiveness of a disinfectant on an inanimate surface. So the other one, obviously, you're testing against microorganisms. And this one, you go through a procedure of where you take a stainless steel cylinder, you drop it in a microbial culture, let it dry, and then the cylinder is dipped in a solution of disinfectant at different concentrations. And then you place the cylinder also in a sterile medium without the disinfectant and incubate it, and you measure the amount of turbidity. If turbidity is present, that cloudiness, that means that the microorganism grew. If turbidity is absent, that means there's no growth, so there's no microorganism there. So these are ways of controlling the growth of microorganisms in our environment. We're not talking about in the body or on the body. We're talking about on the surface as far as antiseptics. But we're talking about mostly, in the environment.